Most welcome in my channel. We are going to just a short summary of Lac Operon models, how they work, how they can start it. You know there is a three structural genes are presented. These are structural genes. The name is called is the ZYA, and this is the promoter and operator. This is just a framework of the gene where we are going to understand. In this Lac Operon model is depending upon the lactose. If the lactose is absent, the Lac repressor, this is the repressor molecules, they can bind with the operator molecule. So the RNA polymerase is not able to process their functions. So structural genes are stopped, they are not showing any type of the functions. And when the presence of the lactose in that system, Lac repressor molecule bind with this lactose. So in this diagram you can see this repressor molecules bind with this lactose, and the RNA polymerase now free to operate from promoter to another structural genes. These structural genes now ready to their functions. They are start doing their functions. They are going to translate their proteins for their required proteins formations. So this is the process where the lactose are present or lactose are absent in this Lac Operon is depending upon that the whole system is work or not. If the present, they work. If there is no present of lactose is there, this repressor molecule bind with the operator molecule and the process will be stopped here. They are not going to stop functions. Another two factors are also not only for lactose, cyclic AMP plus cap complex also stimulate polymerase, RNA polymerase activity, increase their synthesis. They can bind with the promoter side, while lactose can bind with the repressor proteins where the repressor are presented. They are binding with the operator regions, so they can bind here and remove the repressor molecules from the operator. This RNA molecules free from that. But in this process, cyclic AMP and cap proteins are also promote the RNA polymerase activity by binding with promoter region and give the signals for Lac Z, Lac Y, Lac A proteins for their proper functionings. So you can understand how the Lac Operon can be promoted, can be stopped by using lactose or cyclic AMP and cap molecules is here. But if there is a repressor is present but no lactose is present, so the cyclic AMP cannot process their functions. This is one of the most important point you can understand. If the lactose is not present in the system, cyclic AMP and cap molecules cannot promote their function properly. So these proteins are not able to function because repressor molecules bind with the operator, so they stop this process from here. The RNA polymerase is not able to proceed their functions. They can stop during their transcription translation process. So cyclic AMP, cyclic A is only the bind with the promoter and increase the activity of polymerase, RNA polymerase, but not the whole function. So lactose presence is most important in this process. That's why it is called Lac Operon models. Hope you like this diagrammatical representation of the programs. Thank you so much.